All right. Am I unmuted? Awesome. Okay. Hello, everyone. This is going to be a review of some of the stuff that we went over last week. I'm sorry that I wasn't able to get to things like pedigree, sex linkage, including hetero and homogametism, and a really cool example, or gene dosage and sex chromosomes and how that relates to mosaicism in homogametic individuals for certain species. Um, yeah, I'm sorry I wasn't able to get to those. I definitely can upon request. I felt like it would be kind of moot to do it now because that's exam one content. Excuse me, but I could definitely do it for review for the final exam um, upon request. And I actually did this review yesterday. It was like 2.5 hours long. But I was messing up a whole bunch and my examples were really bad. And then my computer died when I, before I even finished and I had to go to class. It was kind of a mess, but here we are. And I think we can get started. No one else is present in this meeting but me. So if you are here, it is because you're watching. Thank you. And if you're here, you are able to navigate to this part of our Blackboard course. And there was a lot of, or excuse me, in the exam reflections, there were a lot of requests for more practice problems to work through. And I understand that that could have been a great boon. There was a great boon to me when I was a student, just practicing through a bunch of different problems. And Manor was actually gracious enough to not only make some practice problems, but also post guides for how to solve them per exam. So definitely check those out. You'll find those on our Blackboard page in the LA Resources tab. So, yes. So what we're doing now is non-Mendelian inheritance, because we are so familiar in genetics with, one gene coding for one trait, and it's very nice and clean. But in reality, that is so little of the genome that actually does that. And there's a lot of funky business going on. Things like cytoplasmic inheritance and the maternal effect, whether or not a gene is even expressed in an individual as we see it throughout populations, especially epistasis, and some funky business with how chromosomes interact. And we will get to all of that today. So, excuse me, getting started. In a tropical wet forest, a species of scarab has evolutionarily developed a gene that codes for the production of a very waxy epicuticle from their epidermis. Okay, that can seem like kind of a jumbly mumble, so I will explain that. I also had a cool example yesterday, but it was a really bad example. So arthropods, they have this kind of epidermal structure where they have their actual epithelial cells. Like this would be our the basal lamina of our skin cells that would go on to produce those keratinocytes that shield us. And instead of doing that, insects, they have their actual cells that secrete layers of material that have different purposes. We have an uncalcified layer that's kind of spongy, absorptive, a calcified layer that is provides strength, a pigmented layer that protects from UV radiation, and an epicuticle that is waxy. And this is important for insects because when you're that small, water tension, or I think that's the word. Yeah, water tension is a deadly force. It can envelop you and trap you. So what lets us swim would actually trap and drown most insects. So having this waxy epicuticle that repels water is really important. And so we have a species of scarab that has evolutionarily developed a gene that codes for the production of a very waxy epicuticle. However, these beetles also, oh yeah, scarabs are beetles from the family, or no, not the family, the order Coleoptera. These beetles also have a gene that codes for immune cells that target the epidermal cells that secrete the cuticle. So they have some kind of autoimmune function, which in this case could be a disease that attacks these epidermal cells and prevents them from secreting these layers, including the very waxy epicuticle. So that's interesting that we have one gene, the gene that produces these autoimmune cells that can like prevent the expression of another gene. Very interesting. Um, so the allele C, capital C, codes for a very waxy cuticle. Oh, sorry, I didn't change it. Oh God. 1 second, please. Yeah, Oh, and as you're watching this, I will ask questions all throughout, and I invite you to. One moment, please. I'm sorry. I have to connect to the internet all the time. But that's not a bad problem to have. I'm grateful to have internet. Oh yes, since you're watching this, there's no one in this session with me. So the only way you know that this exists is that you're watching it on Blackboard. Is I invite you at every point where I like ask a question. It may seem rhetorical, but I am inviting you to answer. I invite you to pause the video and think about it, maybe work through the problem, or just give your best guess, and then replay the video. Obviously, you can rewind, watch it at 2 times speed, do whatever you want. Anyway, yeah. So back to our beetles. The allele capital C codes for a very waxy cuticle, where lowercase c codes for a typical cuticle that isn't very, very waxy. It's just, normal waxy. And then capital I codes for autoimmune cells that destroy the epidermal cells, where lowercase I codes for a non-hostile autoimmune cell. So if these two genes assort independently and a homozygous recessive scarab is crossed with a homozygous dominant scarab, how many of their offspring will have a very waxy cuticle? Okay, before we move on, let's break all of this down. This was already a long question. So where do we start with this? So we have 2 beetles, a homozygous recessive and a homozygous dominant. And we have our genes. So we have a homozygous recessive beetle, and we know that the recessive alleles are lowercase. So we have the first beetle is lowercase c, lowercase i, lowercase I. Sorry. And then we have another beetle who is homozygous dominant. So they are uppercase C, uppercase C, uppercase I, uppercase I. Yes, I write my I's the same. And they cross. So we have our parental generation here, and they will cross and produce their offspring, which we know is the F1 generation. And so when we look at these individuals, this homozygous recessive beetle, they only have a lowercase c to give. They only have a lowercase c, and therefore that is the only allele that they can pass on to their offspring. Same with the I locus. So they will pass on this C lowercase I allele. And the same goes for a homozygous dominant parent, except they have the dominant alleles. So they will, for the F1 generation, provide these alleles, resulting in such offspring. And I only now realize that the letter choices I used were a bad example. So anyway, because these were the only gametes, the only alleles that these parents could have possibly passed on to any of their offspring, all of the individuals in the F1 generation will have this genotype. So let's correspond this genotype to a phenotype. We know that, so capital C is very waxy. Capital C, or C stands for cuticle, and lowercase c is typical. A typical amount of wax in their waxy cuticle. And we have uppercase I that codes for these autoimmune, or hostile autoimmune cells. Auto is a prefix that means like oneself. So autoimmune. Auto means within oneself, or like having to do with your own body. And obviously, we know immunity has to do with destroying foreign objects and maintaining a healthy environment within your own body. You know what immunity is. And so autoimmunity is immunity that affects your own cells, oneself, like ourselves, our animal cells that make up our body. And so you think of things like, I'm running short of examples in my brain. You know, autoimmune disorders are things that attack our own cells. If you have vitiligo, then your autoimmune cells mistakenly, or I guess they're instructed, but you don't want them to attack your melanocytes, the cells that create the pigment that gets deposited into our skin and hair. So people have some patches of darker skin, some patches of lighter skin, and that lighter skin is lighter because their autoimmune cells destroyed the cells that actually produce the pigment. I could have used that as an example. What was I thinking? Anyway, so capital I is hostile autoimmune cells, and lowercase I is non-hostile or typical. You know, it's probably typical because organisms who get attacked by their own immune systems probably don't survive long enough to pass on their genes. That's a generalization, but anyway, so we have a genotype and we know what phenotypes correspond to those genotypes. And we know that capital letter, or we pose that capital letters are dominant to the lowercase letters. We articulated that in the question. Wait, did I articulate that in the question? No, I did not. So yeah, just assume that capital letters dominate lowercase letters, and therefore a very waxy cuticle, or alleles that code for very waxy cuticles dominate alleles for typical cuticles, and so on. So we have in our F1 generation at least one dominant allele at the cuticle locus. And so we have an individual that produces a waxy cuticle. So we can move on to the next locus. And what do we see? We see that this individual has at least one dominant allele at the autoimmune disorder locus. And so even though we have the genetic ability to produce a very waxy cuticle, so we have our cells, just like we saw, we have some layers, and we have our very waxy epicuticle. So even though we have the genetic capability to produce this, the gene at this locus has an effect that overpowers this pathway because it like stops it from happening. If we destroy these, if we have autoimmune cells that destroy our epidermal cells, then there are no epidermal cells to create this waxy cuticle. And so we have one gene that controls the expression of another. And we call this phenomenon epistasis. Epi means on top of, and stasis means like staying the same. And you can think of that as like your chromosome at large, because usually the way we think of Mendelian inheritance is that it stays the same. It's very static in how it transfers genetic information. Obviously, we know that's not really correct, but so we have epistasis here. And the way we can describe the actual genes that contribute to epistasis, these cuticle genes and autoimmune genes, is we describe one of them as being epistatic and the other as being what we call hypostatic. So, oops, that's not even the same color. Haha. One moment, please. So we have a gene that is epistatic and a gene that is hypostatic. I was going to do color consistency, but I didn't want to give it away. So once again, looking at these words, we know epi means on top of, and hypo means lesser than or below. Like if you're hypothermic, you are lesser than or below the thermic amount, the temperature that your body needs to survive sustainably. So we have an epistatic gene and a hypostatic gene, and the epistatic gene sits on top of or controls what happens at the hypostatic, or controls the hypostatic gene. The epistatic gene sits on top of the hypostatic gene, kind of like if you, I think of it as like when you're a little kid and if you have a sibling, then like your sibling is doing one thing, but if you sit on top of them, then you prevent them from doing something else. So yeah, we have our epistatic gene and our hypostatic gene. Our epistatic gene controls our hypostatic gene. So knowing that whatever happens at, or knowing that if we have at least one dominant allele at this autoimmune locus, controlling whatever happens at this cuticle locus, we can say that the autoimmune gene is epistatic to the cuticle gene. So we have our cuticle gene, which is the hypostatic, and the autoimmune gene, that is our epistatic, sits on top of. And so we are asking the question, or we are matching a phenotype to the genotypes, that's right. And so we see that we have this genetic capability to produce our special cuticle, but we have the genetic demand by this epistatic gene that no such cuticle is produced because we destroy the cells that produce that cuticle in the first place. So this phenotype, so the phenotype that is exhibited by all individuals with this genotype, which is all of the individuals possible in the F1 generation, given the parental cross that we had available, is that they all have, I guess, no cuticle, because their autoimmune cells destroy the cells that actually produce the cuticle. So 100% of the individuals in the F1 generation will have this phenotype and this genotype. So how many of their offspring will have a very waxy cuticle? 0%, because 100% of them have no cuticle at all. And now we can move on to the next part of the question. If these offspring cross, how many of their offspring will have a very waxy cuticle? And so we can do some more stuff. What color do I want to use? Let's do that. So we have a cross. If these offspring cross, so two individuals from the F1 generation, two individuals with that genotype, if they cross, what is this cross? This looks kind of familiar, where we have two individuals, 1, 2, who are both heterozygous at two different loci. Interesting. So there are two, like the prefix di, heterozygous individuals. Oops. Oh, wow, this is one of those dihybrid crosses. And we know we get a very beautiful and familiar ratio of phenotypes from this special cross, and that is our 9:3:3:1 ratio, where 9 individuals are nine individuals are dominant at the first locus. Handwriting, I'm sorry. Dominant at the first locus and dominant at the second locus. Dominant at the first locus, recessive at the second locus. recessive at the first locus. Oh, sorry. 9 of them are dominant at the first, dominant at the second. 3 of them are dominant at the first, recessive at the second. Another 3 of them are recessive at the first, but dominant at the second. And 1 of the 16 possible offspring are dominant at the first, or excuse me, recessive at the first and recessive at the second. So now that we know the genotypes that correspond to these individuals, let's match them with particular phenotypes, which we can infer from the information given to us in the question. So once again, we have to investigate the epistatic nature of these genes. And so we see, what color should I use? We see that we have the genetic capability to produce a waxy cuticle, but 9 of the individuals, because they have this dominant allele at the epistatic locus, this overpowers whatever happens at this locus. And so these 9 individuals, we know, have hostile autoimmune cells, and therefore they produce no cuticle. I'll just abbreviate that to be none cuticle. And we can move on to the next ones. So once again, we have the genetic capability to produce a very waxy cuticle because that is the dominant condition. No matter what we have at this second allele, we have at least 1 dominant allele for this locus. And the dominant condition in this case is to produce a very waxy cuticle. And now we can move on to the second locus. And whoa, nice. At this epistatic locus, we don't have any alleles that code for the production of hostile autoimmune cells, and therefore our epidermal cells will survive and be able to produce whatever kind of cuticle is coded for at this first locus. And we see that for this genotype, at the first locus, we have the capability of producing a very waxy cuticle. So 3 of these individuals will have a very waxy cuticle. The other 3 individuals, they don't have the genetic capability to produce the very waxy cuticle, but they can still produce a typical cuticle. They have the homozygous recessive condition, so no waxy, but just the regular waxy. However, at this epistatic locus, we once again have those hostile autoimmune cells that will destroy any epidermis that can create the cuticle at all. So once again, we have no cuticle. And finally, the 1 individual has just a typical cuticle. So let's see whether or not they can actually produce it. And yes, they can. They have the homozygous recessive condition at the autoimmune locus. And we know that indicates, or that will code for a non-hostile autoimmune cells. And so we can produce whatever kind of cuticle is coded for. And in that case, that is just a typical kind of cuticle. Oops. And so we now have the phenotypes that correspond to our genotypes. We see that there are 3 distinct phenotypes. We have the none phenotype. So 9 plus 3 is 12. And then we have very waxy. And then we have typical. So we have this modification to our very nice and memorable 9:3 to 3:1 ratio, and that is the 12 to 3:1 ratio. And this is a condition in epistasis. You've probably been saying it to yourself all along, and if not, it should be evident that since we have the presence of 1 dominant allele that can affect the expression of a gene at a different locus, this kind of epistasis is known as dominant epistasis. Oops. Dominant epistasis, where we have 1 dominant, or the presence of at least 1 dominant allele will influence what happens at another locus. And I had something better to say yesterday that I can't remember. So on exams, I don't know for sure, wink, wink, but you might be asked about this ratio. And I would encourage you to know the ratios associated with different kinds of epistatic conditions. So we know that we just worked it out. A dominant epistasis example will usually result in a nice 12:3:1 phenotypic ratio among the offspring of the dihybrid cross that results from the F1 generation. So the F2 generation will exist in phenotypic ratios of 12:3:1. And we saw that because of the combination of the 9 and 3 into 12. And beside that, it's just our very nice 9:3 to 3:1. I tell you that you might want to expect such a question on an exam that asks, so we have a dihybrid cross and the offspring of that dihybrid cross result in they display phenotypic ratios of 9:3 to 3:1. What kind of epistasis is going on here? Or even what kind of genetic phenomenon is going on here? And you would be expected to say that this is dominant epistasis. So I encourage you to know these ratios and know what kind of epistasis causes them. And we can explore the other kinds, of course. I hate orange. Let's just do black. So we know dominant epistasis is 12:3:1. So let's look at different kinds of, different variations of this 9:3 to 3:1 and what that can create. So if we have, for example, well, I'll just come out and say it, I guess. The other ratios that we could expect, actually, no, let's say, sorry, the different kinds of epistasis. Actually, no, I did this so much better yesterday. What am I talking about? Sorry, I'm bungling this one a little bit. So instead of having an epistatic gene whose presence as a dominant allele can affect what happens at the other locus, the other gene, if instead the epistatic gene could only express its effect in its recessive form, then we would understandably have something called recessive epistasis. And in recessive epistasis, the presence of a recessive allele at the epistatic, or the presence of a recessive genotype at the epistatic locus will affect what happens at the other locus, because we have the first gene locus and the second gene locus. And in the case of recessive epistasis, having a single, or having the recessive genotype will, in this case, let's say the roles were switched and that lowercase I instead was hostile now. So having the homozygous recessive genotype for that locus would produce a hostile autoimmune cells, and that in turn would be what affects whatever goes on at the cuticle locus. So it is only in the case of these three individuals where no matter what happens at this locus, we produce autoimmune cells that destroy epidermal cells, which prevent the creation of any cuticle. And the same thing happens here. So instead of these 9 and these 3 combining to become 12 phenotypically identical individuals, we instead have these 3 and this 1 individual combining to be 4. And that produces our 9 to 3, or excuse me, our 9 to 3 to 4 ratio. There is one more example. And think of this if you have been active in class or even just taking good notes, you'll remember this as the Labrador example, where we had the Labradors who could produce either black or brown pigment. But if we had the lowercase e, then that would prevent, like you could produce all of the pigment in the world that's either brown or black. But if you had this gene, which is the pigment deposition locus, whether or not this pigment is even placed into the hair follicle, then you'd end up with a blonde dog who doesn't have either black or brown pigment in their hair. So yeah, think of that as the Labrador example. And the other one that we explored was not just having one homozygous recessive condition affecting the phenotype. Let's say that if you had either a lowercase I, or a lowercase c, that would also keep you from expressing any kind of phenotype. And you can probably remember the snail example where they started out as, snails with white shells. And by like 2 checkpoints, they could end up producing brown pigment for their shells. So at this first checkpoint, we had 2 things. Oh, and also the genotypes of these snails were like A something, B something. But so considering these snails, they start out with a like whitish shell, and by virtue of their genotype, they can move on to producing a brown pigment for their shell. But if they had in their genotype the a homozygous recessive allele configuration at either locus, it would stop the pathway. So they'd no longer move toward becoming brown. They would be stopped in their tracks and they'd remain as white snails. So the A locus could do that. The B locus could also do that. They could successfully move on to the second checkpoint, but if they had the homozygous recessive condition at this checkpoint, it would also stop them in their tracks and they would not be able to move on to brown and they'd remain white. And so in that case, when we have our 9-3 to 3-1, we have our dominant first, dominant second, dominant first, recessive second, recessive first, dominant second, and recessive first, recessive second. So here, if there is a single locus where there's the homozygous recessive condition, then it prevents the expression of this ultimate phenotype. So here we have no homozygous recessive conditions. So we create a brown snail. Here we have that homozygous recessive condition. So they were able to make it past the first checkpoint, but they got stopped here at the second checkpoint. So they remain white. This happened. Homozygous recessive condition at the A locus, they didn't even make it past this checkpoint. They remained white. And so whatever happens at this B locus, even though they had the dominant condition, they could have made it past the second checkpoint. They weren't even able to make it past the first checkpoint. So they got stopped in their tracks. They remained white. And here, the cards just didn't allow it. They were going to be stopped at either checkpoint, and therefore they remain white. And so if we see the phenotypes that result from this epistatic condition, we see that the 3, and the 1 all have the same, so they combine and number 7. And so we have a 9-7 phenotypic ratio for an epistatic condition where having a recessive condition at either of 2 loci will sit on top of whatever happens at the other locus. And this needing or having like 2 different obstacles to overcome, having a like duplicate challenge is called the duplicate recessive epistasis. All right. So I once again encourage you to memorize these ratios, and even if not memorize them, be able to retrieve them like I'm doing now. It's not terribly hard. You just have to know like what this 9-3 to 3-1 means. I keep repeating it, that it's dominant at the first and dominant at the second, dominant at the first, recessive at the second. So as long as you can do that and make sense of these word problems, then it should be no trouble at all. I believe in you guys. You can do it. And if you have any questions, certainly ask Manara. The practice problems, I'm sure it will help. The ones I mentioned earlier. And yeah, I think that was good for this question. We can move on. Oh, yeah, I was going to do um, the, as an example, the SRY gene and how in humans that determines neonates to, I'm not sure if neonate is the right term. Before you are born, when you are either determined to become a male or not determined to become a male and therefore become or remain as a female, whether or not the SRY gene has anything to do with epistasis. I was going to do that as a special example, but I didn't want to mess it up in case that was wrong. So moving on. Oh, and also, let me see if, yes, I did actually. This example relies on your knowledge of Minecraft. Sorry, so if you've never played Minecraft, it might not make sense. I just thought it was a funky, silly little example. So in Enderman, whether or not Ender pearls break when used results from a genetic maternal effect. So Endermen are these monsters, and they throw what are called Ender pearls, and those help you teleport. You throw them, and wherever it lands, you will travel to instantly. So yeah, and whether or not they break when they hit the ground is apparently controlled by a single gene. And it is possible to have ender pearls that resist breaking when you throw them, so they can be used over and over again. I'm not sure if that's the thing, but I made it up. It's my question. And this genetic inheritance is controlled by a genetic maternal effect in the Endermen and their pearls. So an autosomal allele for breaking pearls, let me get my pen out. An autosomal allele for breaking pearls, capital B, is dominant over the allele for pearls that resist breaking. Okay, so let's write this out. So B is breaking. So we have our pearl and it cracks. And then we have B star, which is non-breaking or break resistant, which I will abbreviate to be, or no, I'll just say break resist. And it is still, shining, gleaming, glowing. It hasn't shattered yet. Okay, so it is an autosomal allele, or an autosomal allele for breaking pearls is dominant over the allele for pearls that resist breaking. So this one is dominant to this one. An Enderman named Elisa has break-resistant pearls and reproduces as a female. Are the following statements true or false? So for this question, you will need to just know what the genetic maternal effect is. If you don't worry. I will obviously walk through it with you. It can be challenging to penetrate at first, but we'll do it together if you don't already know. So pause the video and come back. No. Okay, so Elisa's genotype must be B star, B star. Before we explore that, let's explore the genetic maternal effect. So the genetic maternal effect is all about how the genotype of a mother influences their offspring's phenotype. So usually in Mendelian inheritance, we think like I'm a pea plant, and I don't remember the letters they use, but I have I am round, or I have the genes for a round seed and a yellow seed. So I will produce round yellow seeds. This will be my phenotype. I am the organism with this genome, and therefore I will have this phenotype. However, with the genetic maternal effect, that isn't the case. The genotype of an organism is expressed as the phenotype of their offspring. And that's the only key. So, and obviously, this original mother that we were concerned with passing on their phenotype also had their phenotype passed on to them. by their mother. Their mother had some genotype. Um, so we just have to be able to understand that like the phenotypes created by genotypes skip a generation. It's not that nice, very clear-cut, intuitive Mendelian inheritance that we are used to. So we know the only information that we're given is that Elisa has break-resistant pearls. What is a break-resistant pearl? Is it a genotype or a phenotype? Very good. It is a phenotype. It is the evident characteristic that is coded for by these alleles, these genotypes. So Elisa's break-resistant pearls are her phenotype. So Elisa has these break-resistant pearls. So this is Elisa's phenotype. This had to have been given to Elisa from her mother's genotype. And what is the only, okay, so now that we have that broken down, Elisa's phenotype came from her mother's genotype. So we know Elisa has a specific phenotype. What kind of genotype corresponds to that phenotype? What could Elisa's mother's genotype have been in order to pass on that information and be expressed in Elisa as Elisa's phenotype? So we know that break-resistant pearls are recessive to the dominant breaking pearls. And so in order to be expressed, it needs to be homozygous recessive. So Elisa's mother would need to have been this homozygous recessive genotype, which is break resistance, because break resistance is recessive. So because we were told Elisa's phenotype, we were able to infer her mother's genotype. So knowing that, part A, Elisa's genotype must be B star B star. Is that true or false? I'd say that it is false. Because of this, it's possible that her genotype is B star B star. So thinking of the cross that created Elisa in the first place, we have Elisa's, let's say, father, and we have Elisa's mother. That's a bad M. That's better. So we know Elisa's mother's genotype for a fact because it is Elisa's mother's genotype via the genetic maternal effect, maternal means mother, that Elisa was given her phenotype in the first place. So we know Elisa's mother's genotype was B star B star. It had to have been homozygous recessive to give Elisa this recessive phenotype. Oh, and by the way, this probably looks familiar because it's nearly identical to the problem that we did in class yesterday with Martha the snail. And okay, anyway, yeah, so we know Elisa's mother's genotype, and Elisa is what color? Oh yeah, Elisa's dark blue. So they actually, let me continue that. So Elisa's mother is homozygous recessive. So Elisa's mother only had one type of allele to give, the B star allele. So her mother definitely passed on at least one of those alleles to Elisa. There is no other option. That was the only allele that she had to give in the first place. And so we know Elisa's genotype, sorry, I am not, okay. So Elisa's mother's genotype, her father's genotype, Elisa's genotype. So we know part of Elisa's genotype because we know for certain her mother's genotype, and it was homozygous. So she could only have passed on one kind of allele. Her father, on the other hand, we know nothing about. We were given no information about it in the question, and we don't know Elisa's genotype for sure, simply because of the nature of the maternal effect. Elisa's own genotype is like masked, hidden behind the phenotype that was passed on to her, given to her, demanded by her mother's genotype. And the way that I like envision that is that Elisa has, pretending Endermen are animals um, in the way that we think of them on Earth. So Elisa has a cell. This is one of Elisa's somatic cells, let's say. And Elisa has her nucleus, and she has her, you know, um, ribosomes doing cell stuff all throughout her cytoplasm. But because of the genetic maternal effect, I have color consistency. Because of the genetic maternal effect, the genotype of the mother is like passed on into Elisa's cells. And so instead of paying attention to what's going on in here, the ribosomes, the proteins that are actually like doing transcription and translation, they are using Elisa's mother's genetic information. And so even though Elisa does have her own genotype, Elisa's mother's genotype is what Elisa's cells are using to produce certain cellular products. For example, let's say that your mother had the genotype for, switch colors. Your mother had the genotype for blonde hair, and your father had the genotype for black hair. So when your parents cross and they create you have this heterozygous genotype. So you have the genetic capability for black hair, pretending that black hair dominates brown hair. The capital B allele is dominant to the lowercase b allele. I can definitely articulate that. So you have the genetic capability to produce black hair, because like this is your genotype. So you'd think that this black hair allele would dominate the brown hair allele, or excuse me, blonde hair allele, and you'd end up with black hair. However, just like in Elisa, you have your own genotype and your own ribosomes, but oops, didn't switch colors. Let's make it dark. It is your mother's genotype that your ribosomes are using. And so their cellular products, let's say that this was a melanocyte that is depositing pigment into a hair. Instead of using your DNA to put all of the black pigment into it, pretend that this is black, even though I'm using light green and insisting that a lighter hair color is represented by a darker pen color. I'm sorry about that. I don't think about what I do. So instead of producing this nice black hair, your melanocyte would instead use your mother's genotype and produce blonde hair, which is, I guess, technically the lack of deposition of pigment into the hair follicle. So instead of what you'd think, where you end up with black hair, you end up with blonde hair. Oh my God, it wasn't a good example anyway, so I'm just gonna cut it there. But yeah, you get the point. Moving back to Elisa and Enderman. This is how I think of the genetic maternal effect. And so where was I? Oh yeah, we aren't given any information about the father, and we aren't even given any information about Elisa's genotype. It could be that Elisa's father was also B star, B star, and therefore it could only give a B star allele, resulting in a genotype for Elisa that is B star, B star. But not necessarily. We don't have the information to tell. It could also be that Elisa's father was B star B, and he did give the B star, resulting in a B star, B star. Or once again, because we know that in meiosis, it's for the most part random whether or not a parent gives this allele versus this allele. The father could instead have given this allele, resulting in Elisa being B star. It could have been that Elisa's father was BB and had to have passed on just a B to result in B star B. We simply aren't given enough information. So we cannot say for sure that Elisa's genotype must be B star B star. This is false. Moving on to part B, Elisa's genotype must be BB. Actually, we know for a fact that this is false because of what we know here. Because of the genetic maternal effect, we are able to infer Elisa's mother's genotype, and we know that Elisa's mother's genotype is this way. She had to have passed on, or the only allele that she can pass on is a B star allele. Therefore, it's impossible to not have a B star allele in Elisa's genotype. Okay, all of the offspring produced by Elisa must have break-resistant pearls. Okay, so we are moving on, and Elisa is no longer the offspring that we're concerned with. Elisa is now the mother. Should I erase this? No, I'm not going to erase it because I actually need it. I'll rewrite this first, prepare it. So Elisa's father could have been BB. Let's say B star, B star. B star, B star. Could have been B star B, or, BB star, doesn't really matter. And he could have been BB. So homozygous dominant, or excuse me, homozygous recessive, heterozygous, or homozygous dominant. So all of the offspring produced by Elisa must have break-resistant pearls is the claim. Is that true or false? What color do I want to use? I don't like orange. Let's do lavender. So Elisa, once again, is not the child that we're concerned with anymore. We are now concerned with Elisa's children. And so we know that Elisa's mother's genotype influenced Elisa's phenotype. So now Elisa's genotype will influence her offspring's phenotype. So we are going to have to, in order to find out Elisa's offspring's phenotype, we're going to have to do some investigation into Elisa's actual genotype. So all of the offspring produced by Elisa must have break-resistant pearls. So in order for her offspring to have break-resistant pearls, what would Elisa's genotype need to be? Exactly, just like in the case of her mother, where Elisa's break-resistant phenotype came from a homozygous recessive genotype, break-resistant phenotype must come from a homozygous recessive genotype. So let's see, is this the only genotype Elisa can have? Well, we know that it's not, because Elisa's father could have been any of these genotypes and therefore could have passed on this B star, resulting in the genotype that will be transferred to her offspring. Or Elisa could have been given this one and is now B star, which will be expressed as a breaking pearl, because just regular B dominates B star. So it is not true that they all must have break-resistant pearls, because it's possible that Elisa's genotype doesn't allow for her offspring to produce break-resistant pearls in the first place. So this is also false. I'm going to write that in a lighter color. Okay. At least some of Elisa's offspring must have break-resistant pearls. Is that true? Now you might be thinking to yourself, I don't know, it's possible. But you can look at the question and it's very specific. It's very particular about what it wants. At least some of them must have break-resistant pearls. Not necessarily. It could be that, once again, Elisa's father gave just the regular B, and therefore Elisa has the genotype that would transfer the phenotype of breaking pearls to her offspring. And because of what we explored earlier with, we now have offspring, and I kind of want to get to that with F. Let's answer D and F in the same breath. So let's move backward. I'm going to keep this here though. Oops. Yeah, I'm going to keep this here. So let's answer D and F in the same breath. Oh yeah, I guess I don't have to move on to E first. So if we know that the genetic maternal effect is, actually, no, I want to answer F first. And this is my review. So we're going to do that. All of Elisa's siblings must have break-resistant pearls. Is that true? Let's find out. So we know that the genetic maternal effect, once again, is all about a mother's genotype um, corresponding to their child's phenotype. And because of the mechanism that we explored earlier, where we have, for example, Elisa's cell with Elisa's own genotype, that could be anything. We don't know because of the father. And then we have Elisa's mother's genotype that is being used to actually do the functions of that cell, with regard to the genes that are subject to the genetic maternal effect, because probably that's not every gene in an Enderman genome. But yeah, so it is Elisa's mother's genotype that is utilized for the production of cellular products and for the function of the cell. And because a mother does this for their child, it's not like only Elisa, like Elisa is the only child to have gotten the genetic information from her mother to be expressed as her own phenotype. If Elisa also had siblings, let's say Jeffrey and Josuke, they also have their own genotypes. So Josuke's genotype could have been B star B. Jeffrey's genotype could have been B star B star. But once again, because they all share a mother, and this gene is subject to the genetic maternal effect, it is their mother's genotype that is being used by their cells to produce whatever stuff, in this case, whether or not their ender pearls break when thrown. So because Elisa is break resistant due to her mother's genotype, Jeffrey was also break resistant due to his mother's genotype, and Josuke was also break resistant due to his mother's genotype. They all share a mother, and therefore they all share the phenotype that comes from the mother's genotype. So all of Elisa's siblings must have break-resistant pearls. That is true. And if we look back to question D, at least some of Elisa's offspring must have break-resistant pearls. Color consistency. We have the offspring of Elisa. So offspring. And obviously Elisa's offspring have their own genotype, their own ribosomes, but it is now Elisa's genotype, because Elisa is their offspring's mother, that is being utilized for cellular products and the creation of breaking or break-resistant endopearls. So is it true that at least some of Elisa's offspring must have break-resistant pearls? Well, the phenotype that they express, whether or not that is breaking or break resistant, is dependent on Elisa's genotype. And once again, we cannot for certain know Elisa's genotype. It could be that Elisa's father gave the regular B allele, and therefore Elisa has the genotype for breaking pearls. And therefore, because of the genetic maternal effect, the genotype of a mother influences the phenotype of the offspring. In this case, the genotype of Elisa, who is now the mother, is breaking, and therefore the offspring display the phenotype of breaking pearls. It could be that Elisa got, the B star, and therefore created non-breaking pearls. We can't know, but because we can't know, we cannot say that it is true that it must happen. False. So, part E. Elisa's mother must have had break-resistant pearls. Let's explore that. So we have Elisa's mother. We know Elisa's mother's genotype. And we know that she passed on to Elisa break-resistant pearls because of this genotype of hers. But I said it earlier, just like, what color do I want to use? Fine, I'll do it. Just like Elisa's genotype, or excuse me, just like Elisa's phenotype came from her mother's genotype, I'm going to not use that. Just like Elisa's genotype, oh my God, I'm sorry. Just like Elisa's phenotype came from her mother's genotype, Elisa's mother's genotype, oh my God, I'm so sorry. I will try one more time. Just like Elisa's phenotype was given to her by her mother's genotype, Elisa's mother's phenotype was given to her by her mother's genotype. So we have grandmother. The grandmother of Elisa is the mother of Elisa's mother. Makes sense. And also that should be yellow, but whatever. Ooh, sorry, I've been talking for a long time. So we are only given information about Elisa's phenotype. And from that, we are able to infer Elisa's mother's genotype. But we don't know anything about Elisa's mother's phenotype, whether or not her pearls are breaking or break resistant, and therefore we cannot infer, or, oh God, I got that backward. I'm sorry. Just as the phenotype, just as we could infer the mother's genotype from the child's phenotype, we can use the mother's genotype to infer the child's phenotype, but we weren't given the mother's genotype or the grandmother's genotype. Therefore, we cannot infer this mother's phenotype. So it is not true that Elisa's mother must have had break-resistant pearls. I'm so sorry for messing that up. I'm tired. Gonna take some water. So yeah, they were all false except for part F. Okay, moving on. Okay, so question N, I guess. I also forgot that I didn't number these. I just put N. Anyway, so question N. This relies on something that we didn't do in class. So if you did not watch the videos for the time that we were asynchronous, when Dr. Bell was going to Georgia for her trip, I certainly invite you to learn more about them. Please watch the videos. Also, I have created some videos last semester I'm sorry, I'm not articulating myself well. Last semester, I created some videos going over some of the concepts that we talked about in class or that we didn't talk about in class, such as dominance. And there's one on epistasis. I use, it's in the style of Dr. Bell's lecture, but with some different examples in case you didn't jive with the ones we used in class, and also, my own thoughts and feedback. The dominance, this is a really good review, especially if you didn't like or didn't watch the video, and also sex-based inheritance. And there are some more, but this is in the Discord in the general tab. You might have to scroll up just a wee bit. Yes, I gave it away. This question is about dominance. So I am a kitty cat. Oh, one second. Okay, thank you. Sorry, I had to check my phone. I'm a kitty cat and I have gray fur. My mummy, who's also a cat, has black fur with white spots. In my veins, my blood cells have both A and B antigens, while my mummy only has A antigens. Excuse me. If hair color was controlled by a single gene, what kind of dominance do mommy and I display? What about our blood? So, yep, it's right there in the question. We are dealing with dominance. So, before we get into that, let's just talk about what's going on. So, as we remember from class, we um, hair color, at least in cats, we explored it to be a sex-related or sex-linked gene. And let's pretend that I am a female cat. My mummy is a female cat. These would be our genotypes, two X's, because we're diploid. Also, I did not articulate that for this hair color locus, there are two available alleles. That is the black allele, which codes for the production of black pigment, and the white allele, which codes for the production of no pigment, which shows as a white hair. So, my mummy, oops, I forgot I have to turn that off before I can. So my mummy is a black and white cat. She has several blotches of black hair that also has several spaces of white hair. That's interesting how you can have, if we assume that from what we know about Mendelian inheritance, that typically one gene dominates another. And if the gene is only, or if the trait is only controlled by one gene at one locus, how could you have two different traits on the same organism? That's so weird. How's that possible? Oh my gosh. And so we can probably infer, usually in a question, you would be told that they have the genotype BW, makes sense because they have both black and white fur. And I am a gray kitty cat, like you see here. And I also have this genotype, XBXW. So what's going on? Also, actually, I should articulate this. Let's say that, okay, so Gare Bear is gray and has an AB blood type, which we know from this. We'll explore that more later. And Mommy is black and white with an A blood type. Okay, it's always helpful to write out the phenotypes that you're working with, just so you can keep everything straight. So what's going on here at this hair color locus? What could be happening? Well, I kind of gave it away. It is the expression of dominance. So what happens is that we don't have that traditional Mendelian inheritance where one allele dominates another, because we see on this cat that there are distinct patches of black and distinct patches of white. And here they coexist evenly, maybe not evenly, but they coexist in their dominant state. Because, you know, if black dominated white, then even if you had this genotype, you'd show up as black. If white dominated black with this genotype, you'd show up as white. But you have both black and white. Neither one dominates the other, but they both coexist in their dominant state on a single organism. What kind of dominance have we seen or watched in videos that has to do with this kind of expression? So when I think, or when I see a coexistence, did I spell that right? Coexistence? Yeah. Of two alleles in their dominant state. This coexistence is an example of co-dominance. And this is because hold on, let me zoom in a little bit. Because of, actually, no, I won't get into that, but we see that in some regions, the hair is black. So this allele is being expressed here in its dominant state. And here we have white all around. And it is in this region where the white allele is being utilized, used for instructions on how a melanocyte will deposit or not deposit pigment into a hair follicle, and whether or not it's actually, I'll get into that with me as a kitty. So once again, codominance is the coexistence of two alleles in their dominant state. We have this black allele that shows up as black, nothing else. And we have these white hairs that are the result of the expression entirely of the white allele. And so yeah, my mommy's hair color as a cat in this instance is an example of codominance. Now, concerning me, I'm not sure if I just use cat hair color because I like cats, but I'm not sure if it would be traits that are so similar in cats, like, hair color having to do with, like, would that be able to be two different kinds of dominance? Who knows? Anyway, so this is me. I'm a black cat, or excuse me, not a black cat. I wish I was a black cat. I'm a gray cat. And how could I be gray if the only available alleles are black and white? That just doesn't make any sense. Well, I am sure that it doesn't take a genius to know like how you get gray. Gray is like a gradient from black to white, darker to lighter, and somewhere in the middle you become a lighter and lighter shade of gray. That is pretty intuitive. And so what's actually happening is that this is my genotype. No funny business going on. And both of these alleles are contributing. So within my cell, I have my genotype, and both of the alleles are contributing to my phenotype. And if this is a hair follicle, let's say that this is my black allele that is contributing, and this is my white allele. So my black allele is depositing pigment. Actually, I will actually make it black so that it's not such a silly example where I'm using blue and green when I shouldn't be. So my black allele is providing the instructions to my cell to produce this black pigment. But my white allele here is also providing the instructions to not deposit any pigment. So in some regions, pigment is not deposited. And so as you get less and less black pigment overall, and you zoom out on this hair follicle, it eventually, oh, there's no gray pen color, dang it. It's intuitive. Eventually it becomes, or it appears to be gray. So what is this where we have two alleles that contribute to the phenotype? Two alleles that contribute to the phenotype. And their combination, and when they both work in concert, so two alleles that contribute to phenotype, and when concurrently expressed, so expressed at the same time, an intermediate phenotype, or a phenotype intermediate to the alleles that when expressed entirely are opposites, like black and white is opposite. And when they're both expressed, you get something in between black and in between white. So two alleles that contribute to the phenotype, and when they're expressed concurrently, an intermediate phenotype is, actually, let's reword this. So two alleles that contribute to the genotype, actually, no, that is technically phenotype. But that definition is less clear. Also, I will upload not only this video, but underneath the video, you will see attached, or maybe even above the video, two Word documents. One of them is this review, just the questions without answers, and the other one will be this review with answers typed out. So and I prepared those beforehand last semester, I think. So they should be worded better than what I'm doing now, or maybe not as well, and I'm just discounting myself. So two alleles that contribute to the phenotype, and when, I guess, expressed concurrently, an intermediate phenotype is visible, is expressed. And so just like, black, a little bit of black and a little bit of white makes for gray, when we see an intermediate phenotype that results from the tandem expression of the black and white two opposite alleles, this intermediate phenotype is the result of incomplete dominance. So we have intermediate, incomplete, codominance, coexistence. And that is the kinds of dominance that are going on with my mother and I's hair color. So what about our blood type? I'm just going to get rid of it. So what about our blood type? So I am, it says in the question, my blood cells have both A and B antigens. Antigens are little sugars. Pretend this is an erythrocyte, our red blood cell. They're just little sugars that I think, well, maybe they're not glycoproteins. So I think they do assist in like autoimmune recognition, which we have explored a lot in this review. So we have these little sugars on the outside of our red blood cells, and they're either like the A variant or the B variant of that sugar. And this is what it means to have an AB blood type. I will not comment on the RH factor, whether or not it's plus or minus. Actually, we did that last semester in Dr. Chiari's class, and Sam had a really good example for it. But anyway, so an AB blood type results from the presence of both A and B antigens on our red blood cells. That sounds familiar. The coexistence of two different phenotypes that result in, the coexistence of two phenotypes expressed entirely, like on some unit of an organism. So we probably have an allele that is I sub A and I sub B. This is how we express blood types, I think, in genetics. So we have this A allele that is producing A antigens, and we have this B allele that is producing B antigens in their dominant state. And so a coexistence of traits in their dominant state is probably codominance. Blood type is a really well-worn example for codominance. But what about my mommy? She only has an A blood type. Is this still codominance? Her blood cells probably look like this with just the A antigens on the outside. Well, it is still codominance because it could be that her blood type is like this, where she has one allele that produces the A antigens on her blood, but she could also have this O allele, this non-allele that produces no antigens on her blood, like in this section right here. And even though it's like a non-allele, I'm making that term up. I'm not sure if it's a real part of the genetic terminology. Even though it's a non-allele, it is being expressed because in this section, maybe where we would previously have had this, it is being expressed as nothing, which is what this allele intended. So this is still codominance. It is still the coexistence of two alleles in their dominant state. The mother could also have been IA, and we would only see that as A antigens. I will not comment on whether or not that means you produce twice as many A antigens as this genotype, but you know. So yeah, pretty nice and simple. Okay, moving on. Sorry about that. I think there is something that I wanted to say with that. Oh no, I did. Okay, moving on to question, I think 5 or maybe 4. I don't know. So the Southern Water Tribe. Oh, I knew that. So 1 second, I'll be right back. Okay, so sorry about that. So question 4, the Southern Water Tribe is composed of a population of 1,000 people, of which 80 are waterbenders to some degree. And if you're not familiar with Avatar or The Legend of Korra, this may also be confusing. Sorry about my funky examples. Actually, I'm not sorry. I love Avatar. But if you're unfamiliar, waterbending is a martial art where based on the flow of energy throughout your body, you can manipulate the different elements. And we see here Katara can like bend with several of her different limbs with her arms, her hands, and the master up here can, bend with his feet. So out of these 80 waterbenders, 60 of them, just like Katara and the master, can bend with any movement with their, or be it their arms, legs, hands, or feet, while the other 20 of these 80 can only bend with their hands. So, interesting that they have like a reduced or limited phenotype. Interesting. If all of the 1,000 water tribes people have the genotype that enables waterbending, why might so few display the waterbending phenotype? Do we have any ideas? How could this be? So if they have, let's say that it's controlled by a single gene and they're human diploids, let's say that capital W is the non-bending gene. Their chi paths are not as open, so they cannot bend any elements, and lowercase w allows them to bend water specifically. So if all 1,000 of the individuals in the Southern Water Tribe have this recessive, homozygous recessive genotype, how could it be that so few of them, only 80, have the capability of waterbending? Only 80 can waterbend. This is, should I reveal it? 1 second. Okay, if you were paying attention at the beginning, you heard me actually say it out loud. This is possible because of an idea called, oops, where's the pen? An idea called penetrance. And this is like not what you would think of if you were just thinking about genetics in the usual way. If we think about Mendel, we had, pea plants, and the pea plant could be green and round, green and wrinkly. And if it had the genotype to be green and round, I think those are the letters that I used, versus green and wrinkly. Like if the organism had this genotype, it would correspond, it would produce the expected phenotype, like 1 for 1. Green, round. So you have the genotype, check, therefore you express the phenotype that you expect, which is associated with that genotype. But penetrance is this this last little trick. I don't know if it's if by some chemical mechanism or if it's a quirk of chromosomes that sometimes our genes just don't get expressed in our phenotype. And so we have this population of 1,000 people and only 80 can waterbend. So we have a bunch and only 80, pretend there's a pie chart, can waterbend. So how is it that's possible? The way I think of penetrance is that, going along with the water example, you have like a population. You have a cloth that is made up of 1,000, let's say, fibers. You have a 1,000 thread count cloth. And if you pass water through it, that, let's say, out of this 1,000 fiber cloth, and you pass 1,000 droplets of water through it, if every thread can absorb a drop of water, then you pass 1,000 droplets of water through it. If only 80 droplets of water get through, that means that 80 out of 1,000, or 8% of the original condition, that only penetrated through the cloth 8% of the time. So we have this gene that can be forced onto individuals, like fibers, and only a certain percentage of them will see that condition penetrate through the population. So we have 8% penetrance. I explained that so much better yesterday. And that's what's happening. Interestingly enough, we're using a water example for the water tribe. So we have this gene, the water bending gene, and it only penetrates through the population 8% of the time. No, actually, I gave it away. So if I asked you what the penetrance of this gene was in the population given this information, you would know that penetrance is just a frequency or a percentage of how many times a gene is expressed in a population divided by the total number of people who could have expressed it in that population. Math, 8 out of 100 is 8%. So I didn't ask for it in the question, but I could see an exam question where you're asked exactly like this. There are 80 people who expressed some trait out of 1,000. What is the penetrance of that trait? It could also ask 80 people have a trait, 1,000 people are homozygous or not heterozygous for the trait, and then this number is no longer 1,000 because this, if they were heterozygous, then not everyone could express the desired trait. For example, if it was, I'm sure you can figure that out for yourself. But yeah, penetrance is how frequently a gene is expressed in a population. Yeah, so 8%. But you notice out of these 80 water benders, 60 of them and another 20 of them have a different expression of this trait. So even though they did experience the penetrance, it did penetrate through the population to include the 80 of them. Only 20 of them can't bend with as much versatility as the other 60. How could that be? And this is another quirk of penetrance itself, where there are different degrees to which the gene actually penetrates the population. So you could have been penetrated and an entire droplet came through, or one of the fibers, one of these 80 fibers could have absorbed half of a droplet of water, and therefore only half of a droplet came out, and this number is no longer 80 whole drops, but 79 whole drops and 1 half drop. So this is the idea of incomplete penetrance. Penetrance can either be complete or incomplete. And in this example, let's say you had the 1,000 people. Out of the 1,000 people, non-benders, so you had 920 non-benders. So this was impenetrance. the gene was not able to penetrate through them and be expressed as a phenotype. Penetrance. So this is impenetrance. It was not able to penetrate them. And there were, 60 individuals who were able to bend with any movement, with their hands, with their feet, probably even with their mind. And this was complete penetrance. They expressed the trait entirely. It was able to penetrate their genome and be expressed correspondingly in their genome in the entire manner in which you expect it. But 20 of them were able only to express the penetrated gene in a limited way. They could only bend with their hands. So they, the gene penetrated them incompletely. And a more straightforward example of this is, for example, if instead of a gene for water bending, you had a gene for a third arm, where the impenetrance, even if you had the genotype for the creation of a third arm, the impenetrant condition would be where you just don't have a third arm. You only have two arms. The complete penetrant condition of that would be if you could grow a third arm that you could, like wave around and carry objects with. And the incomplete penetrant would be if you just had like a little nub of a third arm. So you expressed a limited, in a limited manner, you expressed the gene that allows you to grow a third arm, but not all the way, not in the entire manner in which you'd expect. Yeah. I'm not sure if you'd get a question on the exam that just asks, 80 out of 1,000, what's the penetrance? Or if you'd be asked a question like this where it would not indicate that this is penetrance and you'd have to figure it out for yourself. But I thought I would ask that kind of question just so I could talk about incomplete versus complete penetrance, which I'm not sure if we talked about. Okay, and the last question. So 2 individual bryophytes have the genotype JM and JM respectively. There's going to be a lot of J's and M's in this question, and you can read, so I don't need to articulate which one is dominant versus recessive. And oh yeah, before we move on, a bryophyte is just the name for moss. Phyte means like plant. Bryo is literally Greek for moss. And so 2 individual bryophytes have the genotype JM and JM respectively. So let's say that this one is JM and JM. Sorry, please go away. Go away. When these mosses mate, so when they cross, actually, I don't want to do that color. I want to do this color. Oops, that's not even the right letter. So these are our 2 parents. 2 individual bryophytes, JM and JM, respectively. When these mosses mate, they produce 33 offspring with the following, they produce offspring in the following numbers. 33 of their offspring. So when they cross, you produce the F1 generation, which is 33 individuals, JM, 22 individuals are JM, 7 are JM, dominant at both loci, and 3 are recessive at both loci, JM. What percentage of these offspring have a recombinant genotype? So this is like kind of an alien sounding word. What does recombinant mean? And this terminology, recombinant, um, and its companion terms have been a very confusing thing for us as a class. I've received a lot of questions about it. And, it's confusing the way that it's been explained so far. And that is no problem at all. We're here to learn. So recombinant, what does this alien word mean? So obviously re and combine are the root words. So re means like again, to do something again, and combine obviously means like to join together, combine. What is combining again? What is even recombining? This relies on something that I hinted at earlier in the semester in the cell cycles review. So when we are in metaphase, when we're in meiosis, the first stage of meiosis, when we're in metaphase, we have our homologous chromosomes that align in the center of a cell. We remember homologous means that they have the same information. And so, which colors did I use? I'm not sure. Oh, yes, that's right. So let's say that these homologous chromosomes, what does it mean to be homologous? We know that that means to have the same information. Homo means same, log means information. So they're homologous chromosomes. These chromosomes have, the B gene, which is, be creative, Bear, how dense your patella is, or how thick your patella is. So patella thickness gene. And you also have the genes, the A gene, which is like the maximum length of your, I don't know, your soleus muscle, soleus length. And so these two chromosomes, one of which you probably inherited from your mother, one from your father, these chromosomes have the same information, the same genes at the same locations on the chromosome, so that when they go through meiosis 1 in metaphase, they can align together and then be split up evenly so that we ensure, because we have more than just one pair of chromosomes, we ensure that every possible offspring that result from meiosis, when we end up with the 4 daughter cells, that they have like all the instructions that we need, so that we have the soleus and patella gene, so that we also have the eye gelatin gene and the heart rhythm gene, So our homologous chromosomes align in the center of metaphase so that they can be split evenly and partitioned among the daughter cells. But it's not that simple. Well, it is that simple, but our chromosomes do something here, or they can do something here that introduces genetic variation. And we know that they can assort independently. We've explored that a lot, whereby instead of having light blue on the left and dark blue on the right, instead we could have dark blue on the left and light blue on the right. And that would impact, once we go through the stages of meiosis, whether or not you end up with a light blue next to a red or a dark blue next to a red. And that creates variation in itself, and that's independent assortment. But chromosomes can also do another thing that allows them to have a little bit of difference, to maybe introduce that perfect amount of variation that produces an offspring who is, actually, I won't articulate that because we haven't talked about evolution yet. Anyway, so genetic variation, is important. And the way that that can be created is by this recombination. And we call it shorthand crossing over. What is recombining? What is crossing over? So when these chromosomes align in the center, let's get rid of this. When our chromosomes align in the center during metaphase of meiosis 1. They align like gene to gene, one for one, hopefully, and they can just by chance, they don't have to do this, but they can at a certain point, like right here, for example, they can exchange arms of their chromosome, like entire segments of the chromosome, and switch information. So we see that there was previously this recessive allele on this chromosome. This chromosome now has access to a allele that it didn't previously have. So this is some fantastic new genetic variation. And whether or not this happens is whatever chemical mechanism allows this, I'm not entirely sure of. And crossing over doesn't have to happen at all, but it's possible to happen. And this, when like you, as I was doing, hey, get out of here. It is when the pieces detach from their original chromosome and recombine with another chromosome. That is what recombination really is. So recombination isn't actually a very alien word. It is just when the pieces of chromosomes are detached and then reattached. And that introduces genetic variation. So that is what recombination is. But like I said, this does not have to happen. And actually, let me go back. And so if you had your original two parents, and so like the genotype that we are looking at in them is, this first one is A, B, lowercase c, lowercase d, lowercase e, and this one is capital A, capital B, C, D, E. So if these came from your parents, actually, no, don't say that. Instead, say that these could possibly be passed on, because we have the anaphases where this homologous chromosome will be separated, or this chromosome will be separated from its homologous chromosome. So it'll be pulled that way and it'll be pulled that way. And it'll result in a cell with a light blue, and this one will be another cell with a dark blue. And then these will be split apart. Sister chromatids will be split apart from each other. So we are focused on like this guy. You have one here and one here. Sister chromatids will be split apart from each other. We know that happens in anaphase of meiosis 2. And so what we were looking at, this chromosome is right here with our A, B, C, D, E. And so this will get passed on to the child of this cell. This is the gamete that will get passed on. And so if crossing over didn't happen, we'd have this A, capital B, C, D, E. But if crossing over did happen, then we would instead have this A, capital B, C, D, E, like this instead, because we switched these two. And so the parent, before crossing over happens, it would have been identical to the parent, where it was just lowercase a, b, c, d, e. But now if we have this, it is no longer like the parent started as. If crossing over occurred, then the gamete that results would be like this with a capital A. So it is no longer like the original parent. It has recombined. And so it's no longer like the original parent. So we have a recombinant progeny here. This is evident in the fact that its genotype, is not like the original parent was when this was back here before crossing over. And so we can also have a non-recombinant genotype, where, once again, crossing over didn't occur. Whereas just like this, nice and simple. And in that case, we would have lowercase a, b, c, d, e, and that would be non-recombinant. And because instead of memorizing it as non-recombinant, I always like to tell myself that it is parental. It is identical to the parent that we saw before, or if crossing over didn't happen. And so keeping that in mind, what percentage of these offspring have a recombinant genotype? So recombinant is the result of crossing over. Recombinant is crossing over. And the opposite of that is a parental genotype. No crossing over. And they are identical to the parent. And in that case, we can very easily identify them. We know the parent's genotypes. So if crossing over didn't occur, then their genotypes should be identical to the parent. These two are identical, and these two are identical. So these are the two non-recombinant, or I like to say parental offspring. And you'll notice that these parental offspring are the most numerous. 55 of them did not exhibit crossing over, where only 10 of them did. You can tell that these are recombinant because at some point the parents had to have swapped, otherwise, because this individual does not look like either parent. This individual, like say that J was all about the volume of, because these are mosses, they're all about the volume of your little microphyls. So J is microphyll volume. Phyll means leaf, micro means small because they aren't actually vascular. They have really small photosynthetic surfaces. So microphyll volume, and capital J was big, lowercase j was small. And so this individual, the original parents were, oh, also I should articulate that the M, for example, could be light green versus dark green. Capital is dark green, lowercase m is light green. These are the phenotypes that correspond to the genotype. So this parent would have, because of capital J, they would have had larger microphyls. So they would be big and light green. This parent would have been small and dark green. So the fact that we have offspring, or we can look at the offspring, this offspring is also big microfilled with light color. This one is small microfilled with dark color. This individual is big microfilled with dark color. That is unlike either of the parents. This is the genetic variation. We have offspring that are unique from the parents. So perhaps these traits are advantageous in combination or in concert compared to the phenotypes that our parents had. Am I recording? Wouldn't that be so tragic if I wasn't recording? Oh, thank God. Okay, by the gods. Anyway, so yeah, a distinct genotype, or excuse me, a distinct phenotype arises when recombination happens. And this is evident in the recombinant, recombinant progeny who evidently exhibited crossing over. And the same thing here. This was a small and light phenotype. Neither of the parents had a small and late phenotype. These are non-parental, also known as recombinant phenotypes. So why are so many of them parental and so few of them are recombinant? This is what I was hinting at earlier, is that crossing over might not happen at all. Crossing over is actually less likely to happen than just not crossing over. It's easier for things in the universe to not happen than they are for them to happen, or it's more likely that things don't happen than that things do happen. For example, like if you're walking into a field, how likely is it that some random ass dog would run up to you and just bite you? That's pretty unlikely. It's much less likely that if you were walking in a random field, a random dog would come and bite you than it is that if you're walking in a random field, nothing comes and bites you. Also, actually, no, I'll get to that when we get to 3-point crosses. But yeah, so we see more parental offspring whereby the parents did not exhibit crossing over because it is more likely that crossing over doesn't happen. Anyway, this question was just about like demystifying what recombination and non-recombination means. So recombinant individuals are ones whose genotypes and phenotypes are unlike the parents, and non-recombinant, which I call parental, it's easier to keep track of it that way, are ones that are just like the parent, you know, just like the parent. Parental. What kind of linkage is likely displayed? Let's not talk about that. And actually, I purposefully chose this bryophyte example as a bad example. Bryophytes, you may have noticed that there are only two things. You may have thought, why isn't it like JJ and M? That is because bryophytes in their different life cycle stages are either haploid or diploid. And actually only the sporophytes, only the very limited plants that produce spores are diploid because they have the zygote attached. They have the zygote attached, and that spore will fall into the environment, grow up to become the actual moss that we see, which we call the gametophyte. Gameto, like from gamete, because it's just N, it's haploid. And that's the moss that we see. And then it will do some reproduction, and it will form a, once again, diploid sporophyte. Anyway, if you take biodiversity, you'll learn this. So yeah, this was actually purposefully a bad example. So because the mosses that we see are actually this haploid, this N, what does it mean to be haploid versus diploid? Very simply, it means that for all of our chromosomes, we don't have a homologous chromosome. All of our genetic information is represented by a single chromosome. And because this is where J is, this is where M is, there is no corresponding J, no corresponding M, because we're haploid. All of our genetic information is represented by a single chromosome. Well, not like in that sense, it's just that there is no homologous chromosome for us to have another allele. So when these align for meiosis, they don't have a homologous chromosome to align with. And actually, I looked it up to make sure, and bryophyte chromosomes are kind of funky business. It's kind of complicated. Like there are U and V sex chromosomes, and they don't even have that familiar X shape. It's weird. Plants do be weird and stuff. So if you're a haploid, then this example doesn't even work, because if you're a haploid and you don't have a homologous chromosome, can you even cross over? No, there's nothing for you to cross over with. So bryophytes actually don't exhibit crossing over. I don't think it wouldn't make sense. Yeah, and I didn't get into distances and stuff. I kind of wanted to do some like JM where J was here and M was here and whether or not the distance between them was a certain amount. But we will do that in the review for this week that we're currently in, which I will have caught up with finally. So yeah, 2 and 3 point crosses next class, or not next class, but this review. We have an exam on the, I think that, I don't want to trick you guys. I think it is from the 21st to the 22nd. I don't want to fool you and cause you to get a 0 on your exam, so I will check that. It is also available for you to find out. But since you're here, yeah, here we go. Okay, exam 2, October 19th through the 20th. Yeah, I would have fooled you guys pretty hardcore. And that is a Wednesday and Thursday. It will be available. It shouldn't conflict. Actually, I won't comment on that. But yep, we will have the exam review for that sometime, probably next week. And I will also have another weekly review at the end of this week. So yeah, I was kind of limited in what I wanted to talk about with this last question, just because I can expand on it more in the next review. But anyway, I kind of missed out on some of the stuff that I wanted to say. And I've been doing this review for such a long time, geez. And this is the second time I did it. Anyway, thank you for sticking around for so long. And if you have any questions about this, please certainly ask Minar or I about it. And we would be happy to help. We are here to help. We are learning assistants. We're here to assist in your learning. That is like the whole purpose. Otherwise, we're just like coming to school really early on Tuesdays and Thursdays for no reason. So if you have questions, please ask us. We're here to help. And with that, we will see you in class tomorrow. Bye-bye.